Okay folks, so we're going to give an introduction to the production of antibodies for clinical diagnostics, and we're going to specifically focus on the ELISA. To learn outcomes, I would expect you to have an understanding of how polyclonal and monoclonal antibodies are produced. I would expect you to be able to define and explain the range of different ELISAs. That would be the direct, the indirect, the sandwich and the competitive ELISA. I'd also like you to be able to articulate the advantages and disadvantages of different ELISAs, and also explain the concept of lower limit of quantification and upper limit of quantification. So the whole concept of this is that in clinical diagnostics you need to have the ability to measure target molecules, and this isn't an easy task, considering you'll want to be measuring specific targets in complex matrices such as blood, serum, urine and cells. And these matrices have a whole range of vast amounts of different molecules such as proteins, electrolytes and other components. And the difficulty is that you want to try and detect and measure one specific molecule within that matrix, and that's the challenge that's presented to us in the clinical laboratory. And generally speaking, analytical methods such as your mass spectrometries and other analytical methods are extremely specific and very accurate, but they're not high throughput. So it's very difficult in many cases to implement these type of technologies in the clinical lab. And the other thing is, a lot of these technologies require some sort of sample preparation before you use them. So we're going to take you through why using ELISA based technologies is a useful approach. And the whole concept is that the immune system has the ability to produce antibodies that have extremely high specificity for a given target. And it's exploiting this property of the immune system which we're going to talk about. So all of this has led to the development of antibody-based techniques that enable the identification of selected targets. And of course the next thing then is, okay, so we detect the targets, how do we quantify them? And generally speaking, in ELISAs, the quantification will be done through the use of enzyme-mediated activity, or in other technologies you can also have fluorescent tags attached to the antibodies which enable quantification. But getting it right back to the basics, what is an antigen? An antigen is anything that can be recognized by the immune system. Now this can be self, our own proteins, but it can also be non-self, okay, such as pathogenic cell wall components and things like that. Now usually they're quite large, and they're usually proteins, but the one thing to note is that these antigens can contain many binding regions that the immune system recognizes, and these binding regions are called epitopes. Okay, so for instance, in the diagram presented, this antigen has 4 different regions that the immune system recognizes, indicated as epitope 1, epitope 2, epitope 3 and epitope 3, or epitope 4. So generally what happens is, when you get exposed to an antigen like this and you get a B cell response, you'll find that there will be 4 different B cells that produce their own individual specific antibody targeted against an epitope. Now, in a response against this antigen, that means you're going to have 4 different B cells. One B cell producing an antibody specifically for one epitope. Okay, and just move on. Okay, so here we have the antigen, and here we have 4 B cells that are produced as a result of being exposed to this antigen. Each B cell produces its own unique antibody, and that antibody will be directed against an epitope. So for instance, with the bottom cell, the purple cell, it produces antibody that will specifically interact with epitope 1. Okay? And in the second scenario, the blue plasma cell will produce antibody which will be targeted against epitope 4. And then the grey plasma cell will produce antibody direct against epitope 3. And the orange will produce an antibody direct against epitope 2. So that is how the immune system will respond. Okay, and here we have each one binding. So now I'm going to talk about how this can lead to the isolation of polyclonal antibodies. Okay, so we have a target. Okay, so say for instance it is C-reactive protein. Okay, a protein in our bodies that gives us an indication of whether an inflammatory response is taking place. So we isolate a version of C-reactive protein. We modify it to convert it to this thing would be called a hapten. And the basic concept of this is that basically you're modifying the antigen to make it more readily recognized by the host. In this case it'll be a mouse. Okay, so it's ensuring that the target will be picked up by the immune system. So once we've produced this modified antigen, we inject it into the mouse model, and we wait for a period of time, and during that period of time the mouse will produce a cocktail of antibodies directed against CRP, or the antigen. So we allow that reaction to take place, and then we collect the serum and we extract the antibodies that's produced. Okay, now the antibody that's produced will be similar to this scenario where you have different antibodies targeting the antigen, with each one of those antigens targeting their own individual epitope. So the polyclonal, the antibodies isolated are referred to polyclonal antibodies because there are a number of different antibodies produced against different epitopes of that antigen. Okay, so that's polyclonal antibodies. Now, what if we want to isolate one of these individual antibodies, or specifically? So what we do is we go through the exact same process. We modify the antigen, we expose it to the animal. The animal then creates an immune response. And when it creates an immune response, it'll produce the B cells associated with it. And what the research companies then do is they isolate an individual B cell from the spleen. Okay, they isolate an individual B cell that will produce a specific antibody. They then fuse that with a myeloma cell, and the reason for that is in order to expand or increase the number of these B cell clones, we fuse it with a cell that's immortal. Okay, so it's a cancer cell, and when you fuse it with a cancer cell, you get a hybridoma cell. Okay, and that basically makes this B cell now immortal. That means we can then continuously, over a long period of time, reproduce this isolated B cell clone. And we do that through cell culture. And when we produce sufficient numbers of these B cells, we then stimulate them to produce antibody. And the antibody that's isolated is one specific antibody directed against one specific epitope. And that's why it's called a monoclonal antibody, because it's a single antigen specific antibody that we can then use to detect our target. Okay, so the antibody collected at the end of this process is sensitive for the detection of the original target, and in the original example I said CRP. So if someone came into the doctors and they wanted to work out if the individual had in fact had an inflammatory response or had an infection, they would take their blood and use a kit containing this antibody that detects CRP. And that way they'll be able to quantify it and make a clinical decision. Okay, so this is essentially what happens. You have 4 clones producing the antibody response, and when we're isolating the monoclonal antibody, we only isolate one of these. Then we fuse it with a hybridoma myeloma cell to produce an immortal B cell clone that we can then progress to produce a single antigen specific antibody.